Hello aspirants, welcome to Arthashastra IAS Academy. My name is Shailaja Arati. As you all know, a very unfortunate incident occurred in the recent times. Air India Boeing 787 plane crash has happened in Ahmedabad. So you all know identifying the victims and handing over their bodies to the families really became a complex as well as an emotional task for the authorities. In this context, let's understand how this DNA analysis is done. So this class is all about what is this DNA, how the DNA analysis is done, especially in the cases of this type of disasters where earthquakes, like the mass disasters happen, earthquakes, plane crashes, train crashes happens, how the authorities identify the DNA. So, DNA is the genetic material of the human beings. It carries all our inheritance ideology, which transfers from generations and generations. So, in this picture, you can clearly observe the DNA, and this DNA is of two types. So, DNA is of two types. Okay, so the DNA one is found in the nucleus of the cell. This is called as the nuclear DNA, and there is also another DNA material which is found, and that is found in the mitochondria of the cell. Okay, so the mitochondria is also called as the powerhouse of the cell. So we have two types of DNA in our cell. So the DNA, it is called as the deoxyribose nucleic acid. So what is this deoxyribose? Deoxyribose is nothing but a sugar, just like the sugars, sucrose, fructose, and etc. Just like that, deoxyribose is also a sugar. Okay, so it is the hereditary material in the human beings. So next, not only in the human beings, DNA usually acts as the hereditary material, and apart from there is also another hereditary material that is called as RNA. Usually RNA acts as the hereditary material in the virus. Okay, so this DNA contains a very unique genetic codes which are embedded in the form of some codes. What are those codes? Those codes are made up of these nitrogenous bases. These nitrogenous bases are none other than A, T, G, C. So where A stands for adenine, T stands for thymine, G stands for guanine, and C stands for cytosine. Okay. So these genetic codes are different from every individual in the human world. But in the case of identical twins, okay, so we call them as the monozygotic twins. So in the case of monozygotic twins, usually the DNA is almost same. Why? Because they have emerged from the same ovum as well as the same sperm. That is the reason for the monozygotic twins, their DNA is almost same. Okay, so in the case of identical twins, the DNA is almost same, but otherwise, every human beings they have different DNA. But if you're talking about the other biological parameter, which is usually taken into consideration, that is iris, which is present in the eye. Okay, iris is different for every individual human being. Even in the case of monozygotic twins, also iris is very different. Okay, so here we see the identical twins and no two individuals have the same DNA, which we have already understood. Now let us understand about the structure of the DNA. In this picture, you can clearly see what is the structure of the DNA. So it is helical. And what type of helical? So it consists of two strands. Then that is called as the double helix model. So moving on. So now let us understand what is the role of DNA in the disaster victim identification. So as the DNA is very unique, so in the mass disaster events such as the plane crashes, earthquakes, landslides, and etc., where mass number of people are dead, then what will happen? So identifying their bodies and handing over them to the victims is really a challenging thing. So then the DNA comes to our rescue, and it is the most reliable method of identification. So the DNA is taken from, usually from where we do extract this DNA. This DNA is extracted from the biological relatives. Who are those biological relatives? They are the parents, siblings, and children. So because we share that is the children, if they are born to the parents, then what will happen? So those people who has this blood relation, so that is the parents, siblings, and children, from them we can identify the DNA. And of course from the personal items also. So what are those personal items? Toothbrushes, hairs, and razors. So where the cells of the body are identified, in this case also we can actually obtain the cells through which we can do this DNA identification. Now let us understand what are the challenges in this DNA based identification. So one thing is, the first and foremost thing is that once the person is dead, then what will happen? The body start decomposing. So the more days, more decomposed body will be there. So decomposition in the crashes makes DNA extraction very hard. And the second thing is the contamination. So because it is because of contamination also, what will happen? Contamination of various types. Okay, so what will happen? That can reduce the accuracy, because the deterioration of the cells will happen at a very faster rate. And moving on, time consuming also, especially in the large scale disasters. In the 9/11 attacks, it almost took 10 months to identify. Okay, because more number of people are doing applying this DNA method is really very tedious task. So, and next comes the other challenge is the storage. That is, DNA survives best in the bones and the teeth. So bones and teeth, because they comes under our skeletal structure, it is best stored in this particular parts. So they resist the decomposition at a very faster rate. Of course, DNA in these parts also decompose, but at a very slower rate. So now let us understand the methods of DNA identification. So what are the different types of methods which are involved in this DNA identification? The first and foremost method, that is the short tandem repeat. In short, we call it as the STR. See, this particular topic is not only relevant for the UPSC mains anthropology, which is relevant for paper 1, where the applications of anthropology is very important, but it is also relevant for prelims also. Usually this match the following type, in this match the following type, these type of questions are usually asked. So most commonly it is used in the forensics, and we all already understood that forensic anthropology is in part and parcel of our syllabus that comes under paper 1 chapter number 12. Okay? So it combines 15 plus short tandem repeat analysis markers in the nuclear DNA. What do you mean by nuclear DNA? So the DNA which is present in the nucleus of the cell, this is called as a nuclear DNA. So and then the highly accurate. So it is STR is highly accurate and it needs good quality DNA. So next comes in the, that is the mitochondrial DNA. So what we need to understand in this context is that mitochondrial DNA is always obtained from the mother, not from the father. So why it is always obtained from the mother? Let us try to understand. For an example, if you see the fertilization process, imagine this to be the ovum, and then what will happen? So the sperm, it dives all the way and it attacks the ovum. Later, what will happen? So when this penetration happens, ovum will actually take only this part, that is the head of the sperm. Okay, so here if you carefully observe the spermatozoa, so this is how it usually appears. So this is the mitochondrial part of the sperm. So when the ovum takes, what will happen? So ovum will only allow this head part to penetrate into the ovum, and the rest of the part, that is the mitochondrial and the flagella, is completely discarded. So that is the reason in this head part only the nucleus actually appears. So that is the reason. So always mitochondrial DNA will occur only from the mother, not from the father. So here, lot of aspirants in the class, they usually ask me these questions, ma'am. So this will happen normally in the natural fertilization process. What happens in the that is, I mean to say, in the IVF or IUI cases where artificial insemination happens. So where the doctors will allow this entire sperm to enter the ovum. Then what in this case? What will happen? Does the people will inherit the male mitochondrial DNA also? So if that is your question, then the answer is no. Okay, so that is completely against the nature. So usually mitochondrial DNA is usually obtained from the mothers. So then what will happen in the mitochondrial DNA analysis? Usually the female line is taken in under the consideration, that is from the maternal line. So because it is inherited only through the maternal line. So it is used, this method is particularly used when the nuclear DNA is degraded. Then the second method is this particular thing. So it is usually used in the burned or decomposed remains. So and then moving on, Y chromosome analysis. So we already know that. So here there are two types of chromosomes. One is the X chromosome and the other one is the Y chromosome. So compared to the X chromosome, Y chromosome is very short, and Y chromosome is inherited only from the paternal line. Okay, so Y chromosome is inherited only from the father. Mother does never contributes Y chromosome. So in the Y chromosome analysis, only it is taken from the male lineage. It should match with the paternal relatives like the father, brother, uncle, and etc. whom they have this blood relation, consanguineal relationships. And the fourth one is the single nucleotide polymorphism. So what do you mean by nucleotide? Nucleotide is nothing but, so it is a combination of sugar plus nitrogenous bases plus phosphate. So this is a composition of one nucleotide. So in the case of DNA, our sugar is a deoxyribose, it is a deoxyribose. And next we see the nitrogenous base. This nitrogenous bases in the DNA, they are four. Okay, so they are A, T, G, and C, and then the phosphate. Okay, so the single nucleotide polymorphism is taken. Poly means many, many types of changes. Okay, so it is useful when DNA is highly degraded and it detects the changes at the individual base points. So it very individual base points like one nucleotide is usually considered. So it can be matched using some items which are used by the victims, usually like the razors, combs, brush, and etc. They all have some human cells at times. So in the case of burn bodies, usually hair decomposes very fastly because it burns very fastly. So in this case, this type of method is usually taken. So now let us see what is the anthropological importance related to this. We already understood this is part of paper 1, chapter number 12, and the entire, so right from, if you see our syllabus copy 1.7 and from 9th to 12th, these are part of your physical anthropology. Okay, the entire ninth chapter, this talks about the things which happens inside the nucleus. Okay, so it reflects how the biological anthropology, forensic science, human genetics, how they are all intersected, and it shows the real world applications. For an example, the type of dresses you are wearing. Okay, so the type of watches, all these are particularly made based on the sample survey which is collected from large number of people. Okay, they are nothing but the real world application of anthropology in the humanitarian context. And moving on, and highlights the role of anthropologists in the disaster response, like identification, restoration, and so that also, like the human dignity is usually observed even if the person is dead also. So the last rites are to be performed. Okay, we express our deep condolences to all the families who lost their loved ones in this particular tragedy. So may their souls rest in peace. I hope this article helps you in understanding this DNA. We will meet in the next session. Till then, take care and bye bye.